Thank you, Lisa. I'd like to extend my thanks to Mr. Hamilton and Mr. Downing and the rest of the board members for having me and my colleagues here to talk about traumatic brain injury and the work we're doing at the Institute to address it. So what is a traumatic brain injury? A traumatic brain injury is something that disrupts the function of the brain. Common causes are what you would expect: falls, motor vehicle accidents, sports and recreational activities, and in the military, a lot of times you look at blast injuries. So how many people are affected by this yearly in the US? In the United States, about 200,000 hospitalizations and 70,000 deaths occur because of traumatic brain injury. Older adults are overrepresented in this population because as we age, we become a little bit more fragile and we lose our balance more easily. Children also have about 630 traumatic brain injury cases a year, and the leading cause of death of children between 0 and 4 are falls. Men are also overrepresented in this because we do things like this. We're twice as likely to be hospitalized and 3 times as likely to die from traumatic brain injury. This is not OSHA approved, by the way. So as scary as those numbers were, traumatic brain injury is significantly underdiagnosed and underreported. Symptoms are often subtle and delayed and can overlap with other injuries. There's also a stigma in the military and athletics for reporting traumatic brain injury. Traumatic brain injury can radically affect your career progression and how much you're getting paid in those industries. There's also limited screening and evaluation tools. It can be very expensive to go to the hospital, especially if you think that the doctor's just gonna send you home to take an aspirin. And then the way we do triage also leads to this, because if you're coming in to the hospital and you're bleeding or you've got a broken arm, of course those things are taken care of first. But a lot of times there's not the follow-up afterwards to look at if there was a traumatic brain injury that was experienced as well. So why can't we just go home and take that aspirin? Well, the impact of untreated TBI can be significant. You have increasing severity of symptoms like your nausea or your headaches, and it can lead to things like emotional and behavioral changes that can be very significant. The biggest problem, though, is compound TBI. Compound traumatic brain injury is when you experience a TBI while you're still recovering from a former TBI. This can lead to chronic traumatic encephalopathy with CTE, which we've heard a lot about with the football players, and it can increase the risk for things like Alzheimer's and dementia. So the real takeaway I want you to get here is that traumatic brain injury, if it's treated in a timely and effective manner, is a minor inconvenience most of the time. However, compound TBI can be catastrophic. So how do we identify traumatic brain injury? We need new screening tools. So screening is a low-cost, low-effort, and non-invasive way that provides an opportunity for follow-up and early intervention. Anosmia has been identified as a possible screening tool for traumatic brain injury. Anosmia is the inability to distinguish smells or the inability to smell. The assessments for this right now are often subjective. They just ask you, can you smell something? Which turns out it's a really hard thing to answer sometimes. Here at the Institute, we've developed an objective measure for detecting anosmia. So the AMMO screening kit is a detection kit for anosmia that allows you to screen for traumatic brain injury. The AMMO stands for Advanced Military Measure of Olfaction, and the underlying science comes out of the Air Force Research Lab and the Uniformed Services University. They determined that anosmia was highly correlated with traumatic brain injury, and then they contracted with us at the Institute to help them make a device using our medical device regulatory systems. The AMMO features were driven by the intended use in the battlefield. So it's small, it's lightweight, it's waterproof and rugged. It can be administered quickly by people with no special skills or training. It's shelf-stable and inexpensive. And this can be used in a ditch, in the dark, under fire, or in the clinic. The AMMO kit can reliably identify anosmia, which is a strong indicator of traumatic brain injury. So how does this work? First, whoever the medical provider is would open the kit and take out the 6 odorant vials. They would then remove the caps of the odorant vials, crack the vial, and then wait till the tip turns blue. Then they would present it to the person taking the test. The patient would then be asked to identify the smell from 4 choices on the answer card, and then the provider would record that on this card here. After they record it, they would score the kit when it's all done, and scoring poorly on this test is a good indicator that you might need to elevate care or seek medical attention. So what are the next steps for the development in this kit? We need to do some human factors and usability studies, and then we would like to make a 10,000 unit production. We think that'll cost between $500,000 and $1 million to do. Then we would do a clinical trial, which would necessitate a level 1 trauma center and likely take 2 to 3 years and about $5 million. Or this could also go straight to market. The FDA has classed olfactory tests as a FDA Class 2 exempt device because of the low risk, of the low safety risk. So this could be marketed as is currently. Now I'd like to pass it to Mark Liberdoni to talk about his work here at the Institute.
Thank you, Craig. Ladies and gentlemen, good morning. I'd like to continue the discussion on traumatic brain injury, and I'd like to introduce you to the topic of using breath analysis, and specifically the biomarkers that are in your breath, as a possible diagnostic tool for TBI. So as we saw from the previous discussion, the amount of TBI cases is quite alarming. Upwards of 50 million occurrences worldwide per year, and as Craig pointed out, 200,000 hospitalizations alone here in the United States. Now, fortunately, about 90% of those cases are what are considered to be mild TBI. So those are your concussions and your subconcussions. But collectively, TBI is the leading cause of cognitive and behavioral decline. We also saw that the preferred diagnostic tool is neuroimaging, such as MRI or CAT scans. So the challenge is to take the power of a CT or an MRI and bring it out to the field for a field portable diagnostic in situ, sensitive and accurate means of determining TBI. So one of the solutions that researchers and scientists here at the Institute are working on is looking at breath and the biomarkers that are in breath for TBI. So you might be asking yourself, can you really use your breath to determine whether you've had a TBI? Well, there's a couple of favorable attributes to that. First is it's non-invasive. It's very easy to collect a breath sample, and it's very easy to monitor a person's breath in real time. Second, your breath is rich with volatile organic compounds. Literature shows upwards of 1,500 organic compounds are being expressed from your body at any given time. And those compounds make up a pseudo fingerprint for each individual. We all have our own fingerprint in our breath profile, and that's a function of our phenotypic expression. And that's a function of your age, your genetics, the environment, your exposure, any diseases that you have, drugs that you take, the food that you eat, and your overall lifestyle. And that creates this fingerprint that you can actually monitor as a function of time. So you might be asking, is this relatively new? Well, yes and no. Literature shows that breath analysis goes back thousands of years to ancient Chinese medicine. People were actually taught to smell the breath of 6 individuals, and the aroma or the smell that was coming off was correlated to specific diseases or ailments. Fast forward to today, pretty much in the last 50 years, the advancement in analytical instrumentation, and especially computers, has allowed scientists to really dig in and discover the compounds that are in breath and correlate those to specific diseases. So in the literature, it's not uncommon to see breath analysis being used for discovery of diseases, Alzheimer's, Parkinson's, even COVID-19, human performance, personalized medicine, military, forensic, and homeland security, just to name a few. So in a clinical setting, breath can be used to distinguish between your healthy and your diseased or incident state like TBI. You can collect your breath during your normal healthy stage and you'll get your fingerprint profile. If something happens or you become sick, you can collect your breath and you can look at the profile and start to determine what these biomarkers that are changing that are being expelled from your body. The analysis of breath is very sensitive. The detectabilities are very low. They're complementary to blood and urine analysis, which is the normal tool, but the breath is much lower, orders of magnitude lower in seeing the biomarkers that are there. Now, there's a number of methods that you can collect breath. The simplest is just having a patient or a person blow into an inert bag. This is like a Ziploc bag that has a valve on it. You open the valve, the person blows into it, you seal the valve. The breath can now be transported and analyzed in the lab or taken somewhere else. So one example is a project that's going on between Southwest Research Institute and the University of Texas at San Antonio. And the project objective is to look at identified exhaled breath biomarkers for mild TBI. The experimental protocol is listed here. Subjects are wearing a mouth guard that's fitted with an accelerometer. They're exposed to repeated soccer headings. The accelerometer data, along with blood and breath, are collected before, immediately after, and then up to a period of 7 days. The blood is analyzed at UTSA for a single biomarker in the blood that is a known biomarker for inflammation in the brain. The breath is analyzed here at the Institute, where we explore all of the compounds that are in there. We bring all of that data together, and we use machine learning and AI to determine what the biomarkers are that are changing as a function of the brain swelling. So the breath analysis that's done here at the Institute is a method that was derived actually from technology that was designed for monitoring the air cabin air on the International Space Station, as well as the astronauts' breath. We took that technology and modified it to allow us to remove the organic compounds from those collected breath samples, put them onto a sorbent tube, and then place them into the analytical instrumentation. So we heard about GCMS previously. It's a gas chromatograph with a mass spectrometer that allows us to separate all the components and identify each individual one at very low levels. In parallel, material scientists and engineers are working on small miniature sensors that are selective just for those biomarkers that we're finding in these breath samples. So the analysis of breath is very rich with information. This is a screenshot of an individual breath that's being analyzed. Across the top in figure A, we're looking at approximately 250 to 275 compounds. The red signal as a function of time is represented by each spike in the signal as an organic compound. The larger the signal, the greater the concentration of that compound is in the breath sample. The software has the ability to dig into each one of these compounds. Here, the highlighted compound between 11 and 12 minutes is shown in figure B. We have the underlying fragmentation and ionization and mass spectral information, which then the software matches up against the library to tell the analyst what that compound is. Once this is completed for all of the breath samples, the data is then exported out to biostatisticians and computer scientists, where they generate these beautiful visual models and numerical models for all the data. So what we're looking at here now is all of the data that's come before and after a TBI event. We've color-coded the compounds to give you an idea of the family. So we have hydrocarbons in blue, ketones in yellow, aldehydes in green, and fatty acids in red. And we're doing principal component and statistical analysis to determine the amount of variation that are in these compounds. So whether they were present before, not present after, or if they were present in both, were they changing as a function of the incident? From here, AI and machine learning will tell us what the degree of change is, and is that a biomarker for the situation, or TBI in this case. So where do we go from here? Now we're working on taking those biomarkers, combining them with the miniature sensors that the material scientists are generating, and we're trying to put them into field portable handheld devices that you can simply blow into, that are specific for the biomarkers that we're looking for. Again, the goal is to make a rapid, in situ, accurate, and sensitive TBI diagnostic device that's as powerful as an MRI, but much cheaper. So with that, I'm going to turn it over to Daniel, and he's going to continue the discussion on TBI. Thank you.
Thank you, Mark. So while I've really enjoyed working with Craig and Mark and seeing their technology that they're working on, I'm hoping that the technology I'm working on can put them out of a job. And that work is focused on reducing TBIs in the first place, and the specific thing that we're focused on is redesigning and improving the helmets within, or the padding within military helmets. So over the last 50 years, helmets have evolved from being made out of metals like steel. They are now at least 15% lighter, and they have either maintained or improved the performance in terms of stopping different types of threats. In the past, at the Institute, we have developed a human head surrogate, which replicates the human skull, the brain tissue, the skin, the muscles, all in the head. And the reason that we came up with a head surrogate is so that we could study the different effects of different impacts on the head and get real-time data in those events. This is an example of one of those head surrogates. You can see on the image in the middle of the screen the sensors that were fixed to the skull, and then in the bottom right you can see that skull once it's been cast into ballistics gel, and you can see all of the cables kind of running out of the head. Currently, we are in the middle of a Connect program between the Institute and UTSA, where we are working together to improve the performance of helmet pads. It's not to say that the pads in the helmet aren't doing a decent job, but recently, there are lots of new materials and new ways of manufacturing those new materials, and that's what we are exploring in this program. So in this world of sports, as you've seen, helmets are typically designed to stop what we like to call blunt impacts. Those could be falls on the ground, coming into contact with others, think football or maybe cyclists. This is an example of a blunt impact test that was conducted at UTSA. They have a really nice piece of equipment to do this, and it's a very repeatable and reliable way to impact every helmet the exact same way. And this is in slow motion video, so you can see the response of that helmet and that head during an impact. In the military, we still expect the user to potentially have blunt impact cases where they may fall, may hit their head on a wall, there may be a large piece of debris that falls on them, but we also know that they could be exposed to ballistic impacts. Those are typically from bullets or high-speed fragments. This is actually a video of the human head surrogate in action, the helmet in place. The threat that's being shot at it was overmatched, meaning we knew that the helmet wasn't going to completely stop it, and this was going to be the last test for this head surrogate. Lots of damage. And the third thing for military helmets that we can expect the user might come into contact with are blast impacts, which are typically from IEDs or explosives. This is a video of a representative blast test. We have a test dummy standing in front of one of our shock tubes. Our shock tube generates a blast wave without using explosives. So it lets us do a lot quicker testing without having to use explosives all the time. And you will see that the helmet pad, or the helmet itself, didn't really stick around for long. So you may be wondering, how are we going to improve the padding itself, right? These are some pretty drastic threats. Softer pads typically reduce the blunt impact force a little bit better. This is something you would find, again, in those sports helmets. Think a softer kind of foam like you can see here. Stiffer pads will actually help reduce the ballistic force, so think rubber. But then the problem becomes, well, okay, we have a helmet pad, but we needed to have both of these features. And lastly, the outer shape of the pad itself, not so much what's on the inside, actually helps to reduce the force that's experienced from blast impacts. And so our current efforts are designing an inner pad structure that can actually behave in both ways, just like the soft squishy foam, but also it can behave like the hard rubber, depending on the impact that it's exposed to. This is a video of the inside of a helmet during a ballistic test, and you will see just the threat that we're up against and kind of what we're having to design for. In the future, once we have figured out the internal workings of the pad, we will combine that structure and we will basically turn it into a pad that has an external geometry that will help reduce the blast forces. And with that, I will turn it back over to Craig to talk about our brain health working group.
Well, thank you, Daniel. And if I do end up out of a job, maybe I can come help you put those dummies back together. So we've talked now about 3 different initiatives here at the Institute, but in reality, there are a whole lot that are working on traumatic brain injury and brain health. So how are we bringing those projects together in a collaborative way? So we've stood up the Brain Health Working Group, which looks to kind of have a cross-discipline collaboration that addresses traumatic brain injury and brain health. We want to be able to create multifunctional tools and multifunctional and multidisciplinary tools and resources for brain health research and development. And we want to be able to create an institute-wide support network for business development in brain health and TBI. There are a lot of groups out there wanting to work on traumatic brain injury, and we think we'll be more competitive and a better resource if we work together rather than separately. So kind of the high level, Prativa talked about machine learning in healthcare earlier. We want to be able to integrate some of that technology into what we're doing as well. So we're going to be trying to develop a institute platform technology for traumatic brain injury assessment. So we want to use machine learning as an assessment tool where we can take data from a lot of different sources and combine them into one tool. For instance, the screening tools from like the ammo test kit we talked about. Biomarkers, so like Mark talked about, his breath-based biomarker. Sensors, maybe sensors that could be integrated in Daniel's helmets. discrete measurement tools like EEG or EOG or gait analysis from the markerless motion capture technology that's been developed here at the Institute, as well as traditional diagnostics like CT and MRI. We want to be able to take data from all of those sources and integrate them into a tool that gives you actionable data for the patient and the provider. So how would this be valuable? It would allow us to find new patterns across multiple tools that are normally not related to each other. That's really one of the big powers of machine learning and AI. We also want to be able to provide objective data in a field that is rife with subjective data currently. We want to be able to integrate this with electronic charting and patient tracking systems to provide continuity of care for providers and patients across the systems. This also is something that would be continuously improving. Machine learning gets better as you add more data. The more people we can get enrolled, the more cases we have, the better the predictions that we're going to be able to offer. And we also want to be able to include new tools. So not just things developed here at the Institute, but third-party commercial entities as well. We want to be able to develop an adaptable platform that provides information that's accessible, relevant, and understandable quickly and reliably through all phases of care. And that's the real value of what we're wanting to do here at the Institute. We want to be able to create tools and systems that drive better health outcomes for everyone, for the entire community. So that's it, and we really appreciate your questions at this point.